Welcome to the Friday session, the penultimate session of what's been a tremendous meeting. And this morning, we are really delighted to welcome two fantastic keynote speakers, Edith Heard and Michael Elevitz. And the first of the two that's going to speak is Edith, and it's my pleasure to introduce her. So Edith started her academic career as an undergraduate in the natural sciences with a focus on maths and physics before being seduced by the range of open and intriguing questions in biology, which led her to read for a PhD at Imperial in London. And whilst working on her PhD, in her own words, she stumbled into epigenetics when trying to cut DNA using restriction enzymes and being frustrated by the presence of methylated regions. The study of the epigenome became her passion, fueling her career as a postdoc in Paris at the Pasteur. And indeed, there is the dog, Edith. Bang on cue. Now, Edith does have a direct link to Cold Spring Harbor since during her postdoc, she spent a year working at the labs, which she describes as one of the most interesting of her life, learning not only about the cutting edge techniques that were being pioneered at the lab at the time, but also doing so while working, as I understand it, in a converted sheep shed, which is something I didn't know before. Now, using all of the skills that she learned, Edith started her own lab at the Curie subsequently, becoming in 2010 department head of the Genetics and Developmental Biology unit. And during this time, she and her lab have performed pivotal research in the area of epigenetics, focusing particularly on X chromosome inactivation, for which she has rightly received numerous plaudits, including Fellowship of the Royal Society, international membership of the US National Academy of Sciences, and a professorship at the Collège de France. Also, perhaps more impressively than these accolades, she's the founder of an organization that helps support scientists displaced by war or conflict. Now, in 2019, Edith took on a new adventure as the Director General of EMBL, the European Molecular Biology Laboratory, demonstrating her commitment to the international nature of science and also enabling her to push her exciting research vision, embracing the heterogeneity of the environment and pushing molecular biology outside of its classical laboratory setting. Now, for her presentation today, Edith has told me that she's going to talk about new and unpublished work. And when preparing this introduction, I spoke to a mutual friend of ours, Wolf Reich, who told me that when he'd previously mentioned to Edith that he also quite liked to talk about unpublished work, she retorted that it could both be unpublished and quite possibly unpublishable. However, I'm certain that this isn't going to be the case today, or at least I very much hope not, Edith, and that we can all look forward to a tremendous presentation. And so it's my real honor to welcome Edith Heard as the first of our keynote speakers this afternoon. So over to you, Edith.
Thank you, John, for a wonderful introduction. And I'm sure I didn't say that to Wolf Reich. And I just want to clarify that I spent my year at Cold Spring Harbor in a goat shed. It wasn't a sheep shed. It's called Cairns, and that's where David Spector's lab was, and it was wonderful.
I blame Greg Hannon for the misinformation.
But then they had a few sheep there as well. It was for the antibodies anyway. So thank you so much, and I'm really honored actually to be a keynote speaker. When initially I was invited, I thought, why did they invite me? Am I really a genome specialist? And I thought, well, the X is pretty cool. It's a big chromosome, so why not? So I accepted. So here we go. I'm going to tell you about X inactivation, which I'm sure I don't really have to introduce in too much detail. I just want to highlight my favorite bits of it before I dive into data. But I've been working on this process for decades. It feels like one has fun and time flies, but nevertheless, it is a process that has fascinated me and many scientists for a long time. And it's a process that, of course, allows for dosage compensation to be achieved between females and males. The X is big in humans and mice, over 1,000 genes. Many of them are really critical genes, transcription factors, housekeeping genes, chromatin factors. So it actually does matter whether you have two of them or just one. And although the Y is also important, it has very few genes, and they're only important for some special things that males do. But the X, as I said, has many important genes, and a double dose is actually problematic for many of them. And in fact, if there isn't dosage compensation, at least in mice, there's very early embryonic lethality. So it was Mary Lyon in 1961 who came up with the hypothesis that X inactivation actually involves the silencing of one of the two X's randomly and that this state is then stably propagated through divisions right through to somatic tissues and maintained with aging. And so this random process leads to cellular phenotypic variation within the same individual. And so here I'm just showing you some of the examples that she had worked on to come up with this hypothesis. For example, in the mice, the tabby locus, which when heterozygous, and it's on the X chromosome, leads to these variegated coat color patterns. And so you can even see it for some of the X-linked genes that encode for proteins involved in human skin and sweat glands. And this is a sweat gland disorder here. And here I'm just showing you a fancy fluorescent version of all this, where a housekeeping gene expressing GFP or tomato in a female pup shows this these beautiful mosaics. So every female is mosaic, and this mosaicism actually does have implications for disease as well. And I'll come back to that later. So what do we actually know today about X inactivation? I would say a lot more than we did, when Mary Line discovered it, but still relatively little. And I hope I'm going to take you a journey into the sorts of answers that we've been getting over recent years, which have become increasingly more mechanistic and less descriptive. So for a long time, we just described the process in every way possible. And one of the things that was discovered in 1990, in fact, just after I started my postdoc, by a couple of labs, one in the UK, one in the States, was this fascinating molecule known as Exist, which is a non-coding RNA that coats the X chromosome in cis, and you can see here by RNA fish, this beautiful cloud of RNA, this image was actually taken in David Spector's lab almost 20 years ago. And so Exist RNA coats the chromosome and triggers gene silencing. And it's expressed monoallelically only from the chromosome that will be silenced. So I could give you a whole lecture on how Exist is monoallelically regulated, but I won't. I'm going to focus more on how Exist does what it does. And this just shows you that it really is a multitasking molecule. It's retained in the nucleus. It's not very well conserved, but there are a few regions of it that are highly conserved, shown in colors here. And these repeat regions actually do the business, the many different jobs that Exist does. And for a long time, the dogma was that once Exist has done its job, and it shuts down the chromosome, then epigenetic mechanisms kick in to maintain it, such as DNA methylation, chromatin changes, but also shift in replication timing, and also changes in chromosome organization. And for a long time, it was thought that these two steps are almost separate. Exist is required for initiation, and then downstream, the chromatin changes lock in the inactive state. But as you'll see that, this dogma has become increasingly blurred. And so this is another cartoon that sort of shows what we know, or what little we knew up until recently, about what happens during this initial step of silencing. So this is a happily transcribing gene on the active X. And when Exist is upregulated, either during early development or actually in embryonic stem cells, which are a beautiful system, at least mouse embryonic stem cells are, for studying the initiation of X inactivation. So as soon as Exist coats, you start to see a loss of the transcription machinery and a loss of active marks, and then a recruitment of some of the repressive marks that were already known to be involved in gene silencing, such as polycomb repressive complexes, both PLC1 and PLC2, as well as other chromatin changes, such as the histone variant microH2A, and then, of course, DNA methylation, which is actually quite a late mark that locks in the inactive state. And so for a long time, we thought that this was pretty much it. But then a few decades ago now, it was discovered that some genes managed to avoid this process altogether. They just escape from X inactivation. And this has been something that my lab's been fascinated in increasingly in recent years. How do some genes just not do it? And is it something special about the transcription factors that can bind at the promoters? Is it something special about chromatin states, or something about their organization? And so there are these constitutive escapees that always escape, whichever tissue or whatever developmental stage you look at. And then there are another set of escapees that my lab and others discovered more recently that we call facultative, and they only escape in some tissues, and moreover, they do it in a variable way. They don't always escape in every cell, nor in every individual. And so what are these about, these facultative escapees? And again, what are the rules that allow these to escape? And interestingly, many of these escapees, both constitutive and facultative, actually encode chromatin-associated factors, which is one of the speculations that we have, is that maybe you need a double dose of some of these genes because these chromatin factors are actually involved in the process of exonactivation. So basically, what I'm going to try and tell you today about is some of the work that we've done to understand how gene silencing is actually achieved during exonactivation. For a long time, we just knew it was important and that exist was the trigger, but we really didn't know how it worked. And so, of course, that means understanding the partners of the exist molecule. And also, we've explored to what extent chromatin is actually involved in initiation rather than just in maintenance. And this was something that, it sounds sort of obvious, but in fact, no one had really actually gone in and looked at whether chromatin matters for the early stages of exonactivation. And of course, how do some genes escape, as I was saying earlier. So this just summarizes a piece of work that we published a couple of years ago. And this was 2 postdocs in the lab who did an amazing job in a very short period of time, Jan Zilic and Aurélie Bouzard. And they used a very powerful ESL system that we created some years ago, where we can induce exist with doxycycline at will on one of the 2X chromosomes. And we can distinguish the 2X chromosomes thanks to polymorphisms, because these are F1 hybrid cell lines, so black 6, C57 black 6, and Castaneda's chromosomes. And with this inducible exist system, we can always inactivate the same chromosome. Of course, random exonactivation is beautiful but problematic because you end up with populations of cells doing different things. With this system, we could force exonactivation always on the same chromosome, and so therefore we could analyze populations of cells. Of course, one can also do single cell omics, which we have done in the past. It's beautiful and very expensive, so we actually like this system because it's actually quite efficient and cheap. And so with this system, we can then look at all of the chromatin changes that had only really been looked at, gene by gene and by immunofluorescence. So we did really an epigenomic study of many different histone marks. And we also did RNA-seq and TT-seq. So this was, the chip was native ChIP-seq. And we compared then gene silencing using this system. And we created this sort of roadmap of all the epigenetic changes that happen. And I'm just going to show you some snapshots of this. For example, we found that histone deacetylation is a very early event during exonactivation. We looked at different time points of exist induction. So 0, 4 hours, 8 hours, and 12 hours and 24 hours. Gene silencing starts already at 4 hours. And we could see that, and this is looking at the 2 different alleles at the promoter of one X-linked gene, RNF12. And here you can distinguish the CAST and the black 6 alleles, and black 6 is in red. And you can see already between the 0 and 4 hour time points, you're starting to see a reduction in the black 6 H3K27 acetylation levels. So the loss of K27 acetylation is very rapid. And it's faster than some of the other marks that we looked at, including H3K4 monomethylation, which is another enhancer-associated mark. That's one of the slowest, in fact. And we found that H3K4 trimethylation is actually also slower than H3K27 acetylation. So both K27 acetylation and H4 acetylation are rapidly lost soon after exist coats the X chromosome and gene silencing begins. So we tested all the HDACs we could, either with inhibitors or with knockouts. And here I'm just showing you the only HDAC that matters, which is HDAC3. So we knocked out HDAC3, and we found that in the knockout ES cells, gene silencing is actually slower. And we also see a reduction in the loss of H3K27 acetylation and H4 acetylation in these cells upon exist induction. And here I'm just showing you chromosome-wide what this looks like. On the one hand, we looked at ChIP-seq, both in wild-type cells. And here, this is actually showing you allelic ratios. So this is a D-score, which is shown here. It's the number of black 6 reads plotted over the sum of the black 6 and the CAST reads. And the color code is that if you're silencing the black 6 chromosome, basically that's the blue chromosome, if you like. So basically you only see the red, the CAST reads. And so here you can see that after exist induction, within 4 hours, you start to lose HEK27 acetylation very rapidly. And this actually, even by 8 to 12 hours, has hit most of the X chromosome. But in the HDAC3 mutant, you see a much lesser loss of HEK27 acetylation. And this is also paralleled in the RNA sequencing data. But you can probably see that there are quite a few genes that actually still lose K27 acetylation in the mutant and also get silenced. So in fact, only some X-linked genes are really sensitive to this loss of HDAC3. But those genes that are really do slow down their silencing, but eventually they do get silenced. So it seems that chromatin is important at some loci at least, and at this level, for the initiation of X inactivation. Now, the other marks we looked at, I'm not going to go through all of them, but I just want to highlight, we looked at the polycomb marks. So PLC1 associated H2A, K119 ubiquitination, and HEK27 trimethylation. And what we found, to our surprise, was that PLC1 associated H2A ubiquitination is a really early mark. And here I'm just showing you again one gene, RNF12. The active X is in green, the inactive X is in black 6. And you can see that even at 4 hours, we're starting to accumulate H2A ubiquitination over the gene. Whereas the K27 trimethylation, which is PLC2 associated, only really starts to accumulate after about 8 to 12 hours. So PLC2 seems to come downstream of PLC1. But we also found, and this is actually using a mutant that I'm not going to show you here, but we use a mutant where you can still recruit polycomb via exist, but you no longer see gene silencing. And here we found that, in fact, this accumulation of H2A ubiquitination and HEK27 methylation still happens along the X, but never within the bodies of genes. So genes have to be silenced in order for polycomb to spread into them. But at intergenic regions, you still see this increase in polycomb. So this is just a summary of what I've just shown you about the early chromatin cascade during X inactivation, just a cartoon version of it. So when exist is induced, you see rapid gene silencing starting from about 4 hours. You see rapid H2A ubiquitination, particularly in intergenic regions, and you start to see a loss of H3K27 acetylation at promoters and enhancers. And then you see more slowly the loss of H3K4 trimethylation and H3K4 monomethylation. And you see a spread of H2A ubiquitination into genes as they get silenced. And this is followed by the spread of H3K27 trimethylation. So this order, you know, might seem a bit obvious now, but at the time was a bit of a surprise. People had imagined, for example, that PLC2 comes in before PLC1. People had also imagined that, you know, losing enhancer marks such as H3K4 monomethyl would be an early mark. In fact, it's one of the latest. So, so this gave us some indications about, you know, how chromatin might be involved and its importance, but it still didn't tell us how gene silencing actually is achieved so dramatically after exist codes. And a number of groups a few years ago came up with a whole series of studies where they pulled out exist partners. And here I'm just showing you a cartoon from the group of Howard Chang. So this is a Chu et al. Study which we collaborated on with him. And in this study, they used a mutant form of exist for the A repeat region, which is that region that's the most conserved of exist that is involved in gene silencing. And compared it to a wild type exist, and they pull down, using this technique they call chirp, they pull down the factors that are differential. And they found that a few factors seem to be A repeat specific. And I just want to say that this was a unique moment in X inactivation history, because in all of these studies, people seem to have agreed on what the factors that are important for X inactivation are. This is almost unheard of in my field. We always disagree before eventually agreeing. And so here, this is just showing you exist again with its business bits. So I'm just showing you some of the factors here that came out of these studies. And in fact, one of the regions of exist that's known as the B repeat is responsible for bringing in PLC1 via HN-RMPK. I'm not going to show you all the studies that have gone on to prove this, but it's clear that exist does recruit PLC1 via this HN-RMPK B repeat interaction. And this then leads to the recruitment indirectly of PLC2 via the JARID2 cofactor. So this is one part of what EXIST does. It brings in polycomb. The other part of what EXIST does is the silencing part. And here, a number of factors were identified to associate with the A-repeat region. For example, the M6A RNA modification complex, including WTAP and MET-L3, are recruited to EXIST via RBM15. So this has obviously opened up a whole new era and area of research that many people are interested in. I'm not going to say more about that today. I'm going to focus on the other factor which came out in all screens and pull downs, which is SPEN. And so this SPEN factor also binds the A-repeat region, and it's a rather huge protein, almost 4,000 amino acids, and it harbors a number of domains that have been, looked at in some detail in other systems, but it had never been suspected to be involved in X inactivation. So of course it has RNA binding domains, which is how it binds to EXIST, but it has been implicated, for example, in notch regulation and notch signaling. So it has some notch signaling domains, and it also has this domain over here, the SPOC domain, which has been involved or implicated in chromatin and in transcriptional co-repression via SMART NCOR2 and NCOR1. And this, of course, includes the recruitment of HDAC3. So this kind of made sense to us because, we'd already seen that HDAC3 was important. And so this was one of the questions, maybe SPEN is actually silencing via HDAC3. So we got very interested in what SPEN was doing. And an incredibly talented student in my lab joined just at the time that these studies had come out from Howard Chang's lab. And so he decided to set up a Degron system to really explore what SPEN does in terms of X inactivation. And so he used the same cell line I mentioned to you earlier, this XX line with an inducible EXIST promoter, and he knocked in the AID tag, allowing us to use an auxin Degron system at the endogenous SPEN loci. So this is a homozygous knock-in. He also knocked in a halo tag or a GFP tag. We had several lines. And so this allowed him, with this cell line, to both induce EXIST and X inactivation by adding DOX, and to deplete SPEN by adding auxin. And here you can see a Western where with just 1 hour of auxin addition, we actually lose SPEN almost completely. And the beautiful thing about these Degron systems is, of course, they're reversible. So you wash out the auxin and you immediately regain the protein. So this is just 4 hours of washout. So with this system, Francois then looked at what is the impact of SPEN when you induce EXIST. And the bottom line is SPEN massively affects X inactivation. Almost all genes on the X chromosome are affected. So this is a similar map as I showed you earlier. Now it's horizontal. So this is in the control. So basically here you see biallelic expression. When you add DOX and you induce EXIST only, then you get monoallelic expression from the non-EXIST coated chromosome, and that's shown here in red for all these genes. Of course, the only gene that is expressed on the other chromosome is EXIST, and it's shown here in blue. But when you add auxin at the same time as you add DOX, you basically eliminate gene silencing across almost all the chromosome. So SPEN dramatically affects gene silencing. We validated this in a number of ways. And we also looked at a knockout, a mouse knockout for SPEN. And if you knock out maternal and zygotic SPEN, you get the same phenotype as an EXIST knockout. So basically SPEN phenocopies EXIST. And when we looked by live cell imaging and fixed cell analysis, we realized that SPEN is recruited to EXIST immediately upon EXIST accumulation. And so here I'm not showing you movies, but just showing you over time the accumulation of EXIST versus SPEN. And this is using a GFP tag SPEN here, and a knock-in of a BGL tag into EXIST, so we could visualize EXIST with an M-cherry tag. So here you can see that basically both SPEN GFP and EXIST BGL are really co-localized almost immediately upon EXIST induction. So they come on simultaneously, but we could also show that if you deplete SPEN using the Degron system, EXIST is not affected at all in its coating. So this was important. EXIST can coat independently of SPEN, but it can't silence genes without SPEN's presence. So which bit of SPEN is important? So here I'm giving you a rapid overview of a lot of data, but basically Francois knocked out all the different domains that I mentioned to you and checked which bit of SPEN was important. And in fact, only the Spock domain seems to be essential for the gene silencing effect of SPEN. But to show this more directly, he also used the Degron system into which he knocked the BGL motifs into exist. And then he used this to actually tether the Spock domain or other domains to see whether this was sufficient to induce silencing. So with this Degron system, of course, you can deplete the endogenous SPEN, and then he expressed the BGL protein either on its own or fused to Spock and looked to see whether or not this actually rescues the absence of silencing when you deplete SPEN. And so this is what happens when you just express a BGL-GFP protein tethered to exist. So here you see that there's really no repression, but then if you fuse it to the Spock domain, you get back almost complete repression. Not all genes are as efficiently silenced, but there is a vast number of genes who end up being silenced. So this was, I would say, very unexpected that this system would work, but it really did. So basically just the Spock domain tethered to exist is sufficient to induce gene silencing. So how does this work? And what Francois then did was actually take the Spock domain and pull down the factors that associate with it using IP and then mass spec. And what he found was that Spock pulls down a vast number of different complexes. Very specifically, we actually were able to validate some of these. And amongst the complexes it pulls down is NCORE SMART, which had already been shown. And as I mentioned, HDAC3 is part of this, so this made sense to us. But it also pulled down the M6A machinery, which had independently been shown to be recruited by exist-A repeat. So the question was, well, indeed, maybe this is actually via the Spock domain of exist that M6A is recruited to exist. We also found that the NERD complex was recruited by the Spock domain, as well as the SAL4 complex, as well as RNA splicing and localization protein complexes, and to our surprise, transcription complexes, in fact, RNA pull2, and many of its cofactors were pulled down. So this was a bit of a surprise because, you know, this is a domain that is involved in silencing of genes. And here, of course, we were pulling down chromatin remodelers, but also the transcription machinery itself. So Francois decided to look at what this might mean. And the first indication of what was going on came when he actually looked to see where does SPEN go on the X chromosome when you induce exist. And so he induced exist using in the same cell line, and then he did cut and run for SPEN to look to see where does it actually locate across the X chromosome. And what he found was that it actually goes to promoters and enhancers of active genes. Silent genes, genes that are not expressed in ES cells, do not show SPEN recruitment on the X chromosome. But all active genes do, but only at their enhancers and their promoters. And here on the left, you can just see a graph that shows that when you induce exist, SPEN is recruited to the X very rapidly after just 4 hours, but then it seems to decrease. Although it doesn't disengage from exist, it seems to no longer be associated to chromatin, which was a bit intriguing. And so here, I just want to show you several genes that he looked at, just to highlight the fact that indeed you don't see SPEN in the body of the gene, you only see it at the promoter and at the and enhancers for all of these genes. And here you can see what happens after 4 hours, 8 hours, or 24 hours of exist induction. And you can see that there's a rapid increase at 4 hours of SPEN recruitment and then decrease by 24 hours. And in some cases, this decreases to almost nothing. So this means that exist brings SPEN to the chromosome. SPEN binds to active enhancers and promoters, but then as soon as the gene gets silenced, SPEN actually then disengages. So this is a sort of hit and run strategy that SPEN has to induce silencing. And so this association or this link or correlation with RNA pol2 is something that we're very interested in because, Spock pulls down RNA pol2. We see SPEN bound at chromatin where pol2 is present. And we're actually exploring to see how the Spock domain of SPEN interacts with RNA pol2. And in fact, Francois has collaborated with Christoph Muller's lab to actually look at how the Spock domain interacts with RNA pol2 C-terminal domain, and it does interact very specifically with a phosphorylated form of serine 5. And in fact, it's already been shown that the Spock domain also interacts with the N-core in a phospho-dependent manner. So it really seems that Spock might be almost tethered to chromatin thanks to this interaction. The question is this how it's actually also doing silencing? And so if we just look at which other factors correlate with the SPEN peaks that we saw in the cut and run, we found that not only is there a correlation with RNA pol 2 serine 5 for binding, but also with HDAC3, more it enhances than it promoters, and also with MBD3 and CHD4. And in fact, this actually corresponds to the NERD complex, which the Spock domain had pulled out. And in fact, what we were concluding is that SPEN is a key regulator of gene silencing during X inactivation, for sure. It's probably tethered to chromatin via its Spock domain to pol2 and other factors. But on top of that, it is this platform for multiple complexes that can silence transcriptionally active genes. And maybe different genes are sensitive to different components of this SPEN platform, as we like to call it. And here I'm just showing you a comparison that we did using either the SPEN degron, the Spock deletion, or an HDAC3 knockout. And you can see that in terms of gene silencing defects, almost 100% of genes are affected in the SPEN degron. Almost all genes are also affected, the majority of genes in the Spock deletion, but the HDAC3 knockout affects fewer genes, as I already mentioned to you. So it does seem that SPEN really is involved in all gene silencing, but then maybe via the different factors it's bringing in, it has differential effects. And I just want to show you in one slide, one of the factors that we followed up, which is indeed CHD4. And so what Francois did was actually create a degron of CHD4 in the same cell line, in this exist-inducible line. And then he looked to see at what happens when you induce exist and when you degrade CHD4, just as we did with SPEN. And this is a lot of work in just one slide, but what he found is that NERD and via CHD4 does seem to be involved in silencing of some genes. It is important, there is a defect, but only a small proportion of genes. And in fact, it's those genes which seem to be silenced with the poorest efficiency during X inactivation. So the CHD4 dependent genes, X-linked genes, seem to be a subset of genes that seem to be the most resistant to X inactivation. So it really seems as if you do need NERD to facilitate this process. So what we're thinking now is that indeed along the chromosome, probably all genes have a different subset of SPEN's partners or the Spock partners to facilitate their silencing in a kind of tailor-made way. So precision X inactivation, so to speak. So now I just want to finish in the last few minutes. I want to say a few words about how genes escape X inactivation. So when Francois did this experiment, he created neural progenitor cells from the ES cells that were that contained the SPEN degron, so he could deplete SPEN even after differentiation. And what he found was that when you look in these differentiated cells where the X is pretty much inactive, there are a few escapees, but most genes are silent. If you deplete SPEN, not much happens, in fact. X-linked genes are hardly affected. And this is just shown here as cumulative fraction between 0, 24, and 48 hours. You really don't see much effect, except you see a little blip where the SPEN degron has affected a subset of genes. And it turns out that subset is just a very few genes, which are the genes that normally escape X inactivation. And so it seems that SPEN, although it doesn't affect gene silencing maintenance, it does affect the degree to which these escapees are dampened. Even though they're expressed, they're more highly expressed when you get rid of SPEN. So this is just showing you some snapshots of these genes. And it turns out that SPEN doesn't bind the active promoters and enhancers of these escapees as well as it does the other genes that normally get silenced. So it's only there at low levels, but it seems that even those low levels of SPEN that are there can have some effect on the degree to which these genes escape. But that still doesn't tell us how these genes escape or why. And so what I want to just highlight is that these escapees for a long time were thought to be just a bit of an oddity on the X chromosome. And in fact, in humans, it's up to 25% of X-linked genes do show some escape, either this constitutive form or this facultative form. So, And this variability that I mentioned means that females are mosaic, not just because of random X inactivation, but because on top of that, a lot of genes show this variable escape from the inactive X. So indeed, females are even more unique than we imagined. But do these escapees matter? And are they just epigenetic accidents, or are they there for a purpose? So this is still an open question, but I think there's very compelling evidence now that some of these escapees really do matter. So for example, TLR7, which is an X-linked gene involved in the innate immune response, is implicated in autoimmune disease, in lupus. And it's been very clearly shown that a double dose of TLR7 leads to lupus. The more X chromosomes you have, the more likely you are to develop lupus. And this is really linked to this type of facultative escape. So these genes are starting to fascinate many people. And so we've tried to work out a bit more about how escape happens. So here I'm showing you studies from many years ago that my lab published, where we tried to look at how genes behave during X inactivation when exist coats the chromosome. And we showed that although most genes, when they get silenced, are sort of brought into this exist coated domain, they start outside and they're kind of drawn into it. The only genes that remain external to the exist coated domain are escapee genes. So for a long time, there was this idea that maybe escapee genes can somehow loop out of the inactive X chromosome in some kind of 3D way. And that's something that our lab, in collaboration with Job Dekker's lab, tried to explore some years ago, where we did Hi-C, and I'm not going to introduce Hi-C, I apologize. I'm assuming everyone knows what Hi-C is and how to do it. So we did Hi-C that was allele specific to look at the inactive X and how it folds. And what we found was that the inactive X is folded in a very unusual way into two huge megadomains, which are separated by a very unusual region known as DXZ4. But on top of these two megadomains, the rest of the X is fairly boring on the inactive X. It's not organized into local topologically associating domains or loops, except for a very few regions, which correspond to these escapee regions. So here you can see this is the inactive X I'm showing you here with just one little tad here. The rest of the X doesn't have any structure. And this tad-like structure corresponds to one of these clusters of facultative escapees. So we did this by looking at clonal NPC lines where either the black 6 or the cast chromosome was inactivated. That way we can distinguish the two alleles, of course. So this, we've tried to zoom in on this to find out to what extent this unusual structure could correlate with gene expression. So we're doing capture Hi-C, and this is something that we set up actually when we came to EMBL. So we're capturing 3 megabase regions, allele specifically, to look at how chromatin folds within them in different clonal cell lines, because we could see that there was variation between these cell lines in the degree to which there was escape. And I just want to show you a snapshot here of what happens to constitutive escapees. So on the left here, this is the active X for this region that is focused on one of our favorite constitutive escapees called UTX. On the active X, you can see that UTX, which is just here, is present in a nice tad. But on the inactive X, there's pretty much nothing. There's no structure that we can obviously see on the inactive X using this capture Hi-C protocol. So very little structure at constitutive escapees. And this is another constitutive escapee. And you can see here on the inactive X, this is Jared1C. Here it is escaping, and there's very little structure, except when a nearby gene, which normally doesn't escape, or very rarely, HUE1 escapes. And here you suddenly see this tad-like structure pop up. So we realized that indeed you only get this 3D structure when you have clusters of genes escaping. And so we looked at several clones, and I'm going to wrap up. Don't worry, John, I'm almost done. We looked at several clones at this region that shows different degrees of escape. So these are just different clonal lines, and this is the inactive X to show the degree of escape there is. And some clones show very little escape, other clones show a lot more escape. And we then looked at the Hi-C maps that go with. And here you can see 2 clones, one of which shows an awful lot of escape in this region, which is an 800 kb region with lots of genes in it, as you can see here. And it looks almost identical, actually, to the active X. It's very, very 3D structured. But then you look at another clone where there's much less escape in this region. And here you can see there's much less structure. But the structure there is doesn't correspond precisely to the genes that are escaping. So there is some rule that we still haven't worked out about how these 3D structures are forming and how they might correlate with this capacity to escape. So the big questions we have now are what actually drives this escape and what maintains it, and is this linked to structure? And for the sake of time, I'm not going to show you this last snapshot, but we feel that this DXZ4 region, which is the hinge of the megadomain that separates the two megadomains, could be a long-range enhancer-like region that drives facultative escape in a rather stochastic way. And I apologize, I'm not going to have time to show you this, but we made mouse mutants and we see that there is an increase in escape, in facultative escape, when you delete DXZ4. So I'm going to end there with my conclusions. I hope I've convinced you that we still don't know much about X inactivation, but we're starting to understand how gene silencing works. SPEN is a key factor recruited by Exist that brings together with it several complexes that we think are key to differential gene silencing along the X chromosome. It also affects escapees to some extent, and yet escapees can still manage to overcome the gene silencing effect of SPEN and Exist. So how do they do this? For the constitutive escapees, it's still really a mystery, and we're setting up screens to try and understand how this works. It doesn't seem to even be linked to structure. But for facultative escapees, we do think that these 3D structures could have a role to play, either via CTCF type loop extrusion or perhaps by transcription-induced extrusion, and maybe some special factors. And YY1 is one of our favorites because it's actually been correlated to correspond to escapee genes. And I mentioned very briefly that DXZ4 could be a long-range regulator of facultative escape, and this is something that I hope we'll be able to crack in the future. And these are the people in the lab who did all the work, very brave people who followed me from Paris to Heidelberg and who've managed to produce, and this is of course first and foremost François Dossin, who finished his PhD just a few months ago. And I think I mentioned everyone else, but Jan Zilic, who now has his own lab in Copenhagen, Aurélie Bouzard, and also I want to mention Agnese Loda and Antonio, who are both in the lab now, and Isabel Rao, and who have done all the most recent studies looking at the relationship between gene expression and TAD-like structures, and Yuvia, who has been helping to analyze the data, who has her own project on the transcription factors that can be involved. And with that, I will stop and thank you. And I'm sorry to have run over time a little bit.
Thank you very much, Edith. That was absolutely wonderful. Really, really enjoyable. So maybe I'll begin with a question. So do you think you can build a quantitative predictor using the different combinations of binding domains, partners of Spock to predict the extent of X inactivation? Do you think you're now at the point where you really can do that, or is there still information that's missing there?
I think, well, actually, I have a new postdoc, Joyce Mann, who just joined the lab, who's actually going to try and do that. So we want to map many of these Spock interactors by cut and run and see, you know, how well they correlate then with the gene silencing kinetics that we've mapped. So then I think we would be able to start to make predictions. And we actually already did some modeling using other people's chromatin maps a few years ago to try and predict whether any chromatin marks in just using undifferentiated male ES cells, whether any of the chromatin marks there were predictors of the speed of silencing. And we did actually predict in that study that PRC1 bound regions would be rapidly silenced. And that fits really well with what we then saw with the ChIP-seq that I showed you. And we're testing that hypothesis right now with polycomb degrons to see not only does polycomb do something, you know, once Exist is accumulated, but could it actually already predispose genes to be silenced rapidly? So that's a long answer, but yes, basically, yes, I think we will be able to do this, John, maybe with the help of people like you and Ollie. And we would love to be able to do this in the very near future, because we're generating an awful lot of data that I think would allow that.
And before we go to open questions from the floor, so are the binding domains part of being conserved across species here? So the different elements that Spock interacts with, are they conserved across species, or is this very mouse specific, do you think, in terms of the degree of silencing per gene?
So, well, first of all, we're not quite sure where Spock binds elsewhere. So where on the X, it's brought in by Exist. So that's a very mammalian specific process, right, that does it. So no, there's no other system that brings in Spock in such a massive way across a chromosome, and then you have differential effects. But when we did that Spock pull down that I told you about, that wasn't actually in the context of exist. That's just Spock in ES cells. So
Right.
some of the factors that we pulled out there have actually been associated with SPEN's functions in other species, such as Drosophila. And Drosophila SPEN is called split ends, and it has been associated with splicing. So the splicing factors that Spock pulled down, we do think are relevant and conserved across species. But for example, the M6A machinery, I don't think so, to my knowledge. So I think there's going to be some stuff that's conserved and other stuff that's very X inactivation specific or mammalian specific. Yeah.
So then going to questions from the floor. So Miguel Yang, a PhD student from Toronto. So they say, do you think exist is required for the silencing stroke function of SpecSpock, or is it simply needed for their recruitment? Have you tried recruiting SpecSpock to autosomal genes, i.e. via DCAS9?
Yeah, that's a great question. We're trying that now. In fact, we're using, we want to actually silence and activate genes across the X, but elsewhere on the genome as well. And we realize that Spock is our best kind of known silencer. So we are actually doing that. I would have loved, I mean, François almost did this experiment to use some other RNA tethered with BGL to recruit Spock to see if we could do this anywhere in the genome. We haven't done that yet. But indeed, with the DCAS9, we definitely will be able to see how efficiently that works. Yeah.
Excellent. Hao Zhang, a PhD student from Arizona. So, and it's asking, is nuclear lamina involved in X chromosome inactivation?
Well, yes, that's an interesting question. It's been a little bit controversial, but I believe, I mean, the lamin B receptor has been shown to be pulled out also with exist. And Mitch Gutman actually published a paper showing that indeed exist does interact with LBR and at the nuclear lamina, and this could participate in some aspects of X inactivation, but it's certainly not behaving the way SPEN does. So it could be a facilitator, but it's not a driver of gene silencing. And, you know, yes, does the nuclear envelope play any role? I should just say that both the active and the inactive X's like to hang out at the nuclear envelope. So in a way, maybe evolution is kind of helping the X with its need to do gene silencing by making sure that it tends to go to the envelope, because it's very repetitive chromosome, actually. I think that's the simple reason it goes there. But it could be helpful. And indeed, autosomes, when you induce exist on an autosome, it's less easy to silence them. They do get silenced, but one of the correlations that people have made is autosomes that maybe are closer to the nuclear envelope are easier to silence, but then again, it's to do with repeats. The more line-rich autosomes tend to get more easily silenced. So this link with the nuclear envelope is still quite an open question, I would say. Yeah. So
a question from Christine Desteche, you will probably know, from University of Washington. So what do you think would regulate escape in specific cell types? For example, is TLR7 involved? And relate to that, Marichelle Oliva from Chicago, a number of genes seem to escape X chromosome inactivation in a tissue and cell type specific manner in humans. Is this pattern preserved across species? So to the first question, so hi, Christine, I can't see you, but great that you're there. I would love to know, I could ask you the question. Christine is a big specialist of escape, actually. So our speculation for some of these genes is that there is something special about these loci in allowing them to loosen up. And in fact, even though TLR7 is not particularly highly expressed in NPCs, we have found that it's in a region that shows facultative escape. So I think that these regions, which show these TAD-like structures, have a tendency, are prone to allow facultative escape. But then what actually drives it, my bet is it is a transcription factor driven process that, you know, you can, if you switch on if you drive a little bit of transcription of these genes, then they kind of, in a region that can be opened up, they just go for it. And that maybe then trickles into their neighbors. So for TLR7, I don't know which particular transcription factor might do this, but that's where I would put my money. Yeah. And then what was the other question about? It was about the cell types, cell type specificity.
Yeah, well, we haven't done a systematic analysis, but we have looked at some of our favorite genes, like the MECP2 cluster. And I would say yes, from what we've seen comparing human data and mouse data, there is a conservation. But then again, some of these are tissue-specific genes that escape. MECP2 is very highly expressed in the brain, so you would expect it to be to be seen there. But we haven't done a systematic analysis. But my prediction would be that, yes, there is some conservation there.
Wonderful. So there are a couple of other questions, but I think in the interests of time, we should probably move on and say an enormous thank you on behalf of myself and Hopi, Jay and Christina for agreeing to be one of our keynotes, Edith, and pass over to Jay, who's going to introduce the second of our keynote speakers, Michael.